Thanks for the Buck Institute for allowing me to take some time here and talk a little bit about tools and technologies that are really, really critical for the development of some of these technologies that we're all working on for various indications. It's quite an interesting time, no doubt. We've seen some exciting developments occur in the immuno-oncology space, which has really stimulated the cell therapy industry, and I'm going to be focusing more so in the cell therapy space than others, and I think the reason for that is that it's really going to be an interesting opportunity for us to develop technologies and bring them forward. So, as Dave was alluding to, the technologies that are currently under development by tools providers like Thermo Fisher and others in the industry are really becoming accelerated as the need for further definition and further characterization occurs. And the reason for this acceleration in tools development is really to make sure that the filings that are submitted to FDA when new technologies are presented as part of CMC filings, that the data sets that are there are really supportive of the type of therapies that are going on. I mean, we're developing technologies, again, as David alluded to, that are much more complex than therapies of the past. Single, ethical molecules, proteins, antibodies, and now actually living cells that react to their environments. So what I wanted to touch on is basically that the regenerative medicine is going to be part of the future. Cell therapy is going to be part of our future here. The complexity of these types of drugs are increasing, and because of that increase in complexity, we're going to need better characterization and tools to control their manufacturing and understand that the efficacy is going to be critical for future administration. So the manufacturing is quite complex. The processes and materials that are used for the manufacturing of these absolutely affect. Again, this is a living drug that are being put together for affecting biological outcomes. So any component or compound that touches that cell as a drug needs to have very in-depth quality control around it. So the need for innovative tools is pretty self-evident. As we start thinking of advanced treatments in cancer and monogenic diseases and modifying genes, and also, of course, to affect aging, we really need to think about how to best translate those. And translation is really dependent on the materials and processes that are selected to move these forward. So early translation is going to be the key for success, and the ability to progress the realization of this vision is dependent on the selection of tools at an early phase. Again, as David touched on, lockdown of processes usually happens now in phase 2, and if we're using these accelerated regulatory techniques, we want to make sure that the quality of the tools that are going in there are adequate to support that kind of safety profile that's needed to get folks treated with these drugs. So hopefully today, as take-homes, we'll be able to understand the complexities involved with moving cell therapy into the clinic, understand the importance of choosing materials and manufacturing systems and how they affect what you're doing, and then I'd like to get provide some exposure to innovations that will help drive the development of cell-based therapies. So we'll talk about the workflow and challenges, the importance of culture systems, quality, process, and delivery of these therapeutics to patients, and also the characterization tools and gene modification delivery tools that exist today. So here's a slide talking about the cell therapy workflow. It's pretty complicated. You have to obtain cells from a patient. You need to be able to transit those cells to a processing facility. You need to receive those cells into that processing facility. You need to then initiate your work, so isolate the particular cells that you have of interest. In some cases, you want to activate them. In some of these immuno-oncology spaces, of course, you want to engineer them to have receptors that are going to be able to bind to particular cancer types, for example, or to engineer them to affect diseases like Duchenne's, for example. You have to expand those cells. You have to wash them. You have to fill them. You have to freeze them. You have to do testing and characterization. And then you have to still distribute these cells on a global basis. You have to store them, put them in planes, and ship them all over the world. So these poor cells that are sitting in our bodies are removed and are really, really taken through the ringer here. And it's our job to make sure that we have controls throughout this workflow. And that's quite a challenge, really, because anything that happens along this pathway could potentially affect the efficacy of this drug. So very, very important to keep in mind that there are many different opportunities to have things go awry. So we need to really stay on top of all of them. So I'm sure everybody's very well aware of this, and I think the purpose of this slide is really to demonstrate that cell therapy takes a long time. Development it can take decades, if not longer. So a lot of the early discoveries by Carl June at the University of Pennsylvania, elucidated some of these mechanisms of engineering a CD19 chimeric antigen receptor into a T cell, and then have the success that they've been seeing that's now been duplicated at a number of different laboratories and companies, including Kite and Juno, et cetera. But the work that went into that really was pretty substantial. And the breakthrough of the year was in 2013, as elucidated here. And we're probably not going to see some of these technologies until another couple of years, at least probably 2017, 2018 at the earliest. So the development of these technologies really take a long time. And the requirements for commercial success to allow these to be successful and have industrial application and commercial success are pretty deep. So we can use the rule of threes here is where we're going. So for a particular drug to become a commercial success, you have to have efficacy, obviously. You have to be able to manufacture that at a cost of goods that's appropriate from a commercial perspective. And I think Brock was relating to the importance of reimbursement, right? Because unless an insurance company is going to pay for one of these compounds or one of these drugs, it's not going to be a commercial success. So in order to affect those three measures of success, the materials that are going in have to be very well controlled, organized, and understood and managed for safety. So the materials that go into there, the raw materials manufacturing process and characterization tools are all very important for the development of an efficacious and efficiently manufactured product. And as part of the support of the raw materials, you have to make sure that you have the appropriate quality, that they have a regulatory path, and that your supply chain that leads to that selection of that raw material as an ancillary product is there from a manufacturing process, making sure that you have a scalable system, that you have control of that manufacturing process, and if at all possible, to bring that towards automation is going to be very important as you move forward. And from the characterization tools, understanding or having those tools in effect to demonstrate potency, purity, and safety will all be part of the equation for creating an efficacious and cost-effective product that can get reimbursed. So what is cell therapy about right now, and what are the factors that are important in driving and bringing one of these technologies forward? Well, it's really about the biology, right? So the efficacy of your product is ultimately where you want to go. And today, we're looking at a lot of tools that are out there, and media systems, for example, that have been around for quite a while, for a number of decades in some cases. But we're discovering and finding that there are very specific subsets potentially of cells that might have a higher potency than is currently been worked on and been developed. So the understanding of systems biology, metabolic pathways, and signaling molecules are going to drive the next generation of media development and manufacturing processes. And it's this understanding of the biology and getting to a very specific and potent cell type that is going to allow the commercialization of these technologies to move forward. So the biology is ultimately important, but it's also the quality is also very important of these raw materials. So you have to make sure that you have CGMP compliant manufacturing for all the materials that you're using, testing and documentation, sourcing and understanding the levels of animal origin contact, for example, as all these materials are not going into the patients, but they're going to be ancillary materials to the product that goes into the patient. So understanding and working with partners that can provide that documentation to help with your CMC and manufacturing controls, chemistry manufacturing controls section is absolutely critical as you're starting to move forward. And this is really, again, pointed out by David, was the initial selection of these tools and materials is quite critical for early phase development and translation. Then it's also about the process and scale. So developing technologies that will allow commercial manufacturing have to come into effect. So again, media systems, specific process containers, manufacturing systems, bioreactors, and like, all need to be worked with in an integrative fashion to make sure that the endpoint and the efficacy and the biology of the system you're trying to put together will work. So again, the complexity of a living cell and the ability for that cell to adapt makes it that much more critical from a manufacturing perspective to choose the right materials to bring a technology forward. So there's quite a bit of effort that has to go into process and scale considerations. And then finally, it's getting the therapy to the patient. Once you have a drug, a therapeutic, you have to deliver it, and these are ultra cold chain types of logistics that are involved, and right now people are doing extensive work in clinical trial support. There are a lot of challenges out there, and innovation will have to happen in this space as well, as current technologies for dewars and handling of frozen components are probably not going to be able to support the future commercialization of a lot of these technologies. The type of innovations that will have to be brought about, we're not really sure. I mean, it could be from the perspective of delivery, having drones that might deliver these directly, or other types of technologies that I'm joking about the drones word, but other technologies that might relate to alternative technologies for preservation that might be room temperature technology. So these are very important innovations that are going to have to get implemented in the next decades for some of these technologies to be able to be delivered to the patient. So how do you know what happens with your systems, and how do you know when you have all these factors and all these pieces of the workflow interacting on a living cell that itself interacts with the environment it's in? How do you know that you're where you need to be? It's all related to characterization. So the quality is one aspect, but also the ability to measure what you're doing is absolutely critical. So again, applying a lot of the technologies that we're having and continuing to develop new tools to support this in the genomics area, transcriptome area, proteomics, and single-cell biology are absolutely going to be critical to the definition of your product and the ability to bring these technologies forward. Potency assays, development of technologies in this area are going to be critical. Flow cytometry and surface markers can be used. And I'm sure Jean's going to go into a lot of these technologies that she's applying today for her early phase clinical trials in the Parkinson's field. But developing potency to make sure that your cell is going to do what you think it's going to do is very critical. And you can develop potency assays by a number of technologies. This particular example is talking about flow, where immunogenicity is monitored essentially, and that you can look at the effect of an immune suppression by monitoring surface markers through flow. Could be one example of a potency assay, but we have a number of different opportunities to look at technologies for other ways to determine potency. Transcriptome analysis could be very critical too. So looking at gene expression, taking samples from a final product, or normally the transcriptome analysis would be taken over a number of cells. So what you're doing in that sense is getting an average of the different cell types in there. But if you're looking for heterogeneity, you'd want to take a much more smaller sample set. So some of these technologies are now looking at getting very small sample sets. As an example, this gene chip technology can look at as few as 10 cells, so you can get an idea of the heterogeneity of your population and see how that might affect efficacy and clinical outcome. And again, with that type of information, you can then redesign your process potentially to select subsets that might be more efficacious or give you a higher potency. So again, characterization is absolutely critical. Single cell analysis, again, breaking it down and trying to understand what's happening at the cellular level and trying to get away from that overall average group is quite important. So there's a number of technologies that are being developed at Thermo Fisher, but other places as well, of course. Again, taking advantage of looking at gene expression and protein expression as potential effectors of clinical outcomes. Multiplexing assays, using ELISAs, try and get as much information as you can out of a single sample. Of course, these are absolutely critical, again, for maintaining cost of goods. These assays are not cheap to run. So if you can get more and develop technologies, if a tool company like Thermo can develop technology where you can multiplex and be able to run on a fewer sample or a lower amount of sample, but get more information, this is going to be beneficial for the industry as well. Biomarkers, of course, is an area where we'll start being able to really understand what the patient population or who the patients that will respond best to these therapies and what kind of patients will have the greatest response based on biomarker and diagnostic tools. 'Cause, again, this is related to the potential for reducing cost of goods and increasing efficacy for different groups. So determining and distinguishing between responders and non-responders. So a lot of this technology is being developed as well and currently used. Safety testing, at the end, ultimately you have to make sure that your product is clean, sterile, free of mycoplasma, free of endotoxin. So technology is related to that. There are a lot of older technologies, but that are being improved upon, again, from a efficiency standpoint, so that you can do these studies and assays quicker and more efficiently, either using PCR technologies or other fluorescent tests, for example, that are coming out with for endotoxin testing. Cellular analysis, we touched on the necessity for flow in many cases. Technologies are increasing where you can, again, do higher or lower volume sample analysis and get multiple pieces of information out using some of the new technologies that are coming out. And the list goes on. Live cell imaging, automated cell count, high content analysis, again, very important. All of these technologies take a lot of time to develop. They're not they are very important and can be designed to have the greatest reproducibility, but they're not. The development timeline is not the same as developing a drug. So it's easier to develop these types of technologies, but they have to keep up with the pace of desired research. Protein analysis tools, we touched on a number of these: ELISA, multiplex, protoarray, protein based assays, and mass spectrometry as well. Very important tools. Again, all of them being optimized and refined and allocated specifically for the generation and testing of cell-based therapies. PCR systems, genome sequencing, again, to correlate with biomarkers to make sure that we can understand which patients might respond best. It's very important to know what their sequence, their genetic makeup is, so that they can be affected the best. So that's on the tool side, but more from a characterization point of view. But then there are other types of tools that are being generated and used in industry today. So, gene editing, again, very important. Brock alluded to CRISPR Therapeutics, for example, that's moving forward, and we've heard announcements of Sean Parker trying to get into the gene modification CRISPR business very quickly with some activities at UPenn. The understanding of this technology is highly complex, and understanding the critical components and being able to provide the tools that will allow for these gene editing techniques to move forward is very, very critical. I mean, we've got an iterative growth in this market, but now with our next-gen genome editing with CRISPRs, we have to make sure that the technology to manufacture the components that go into that will be of a quality level that will be able to support clinical evaluations. Lentiviral production is another area, again, with an eye towards driving these technologies forward, looking at alternative ways to produce lentivirus for the CAR T space, again, is quite important. So there's technologies that are now being able to get titers that are actually probably twice what we show here in suspension culture. So you're making the processing and manufacturing of these components that go into the cell therapies that much more efficient, thereby cumulatively reducing cost of goods. Again, indicators of technology development as the fields moves forward. Electroporation, a lot of it is very simple at this point, at least from early phase research type devices, but getting this to a larger scale is very actively being pursued as well, to have GMP opportunities, GMP devices that can provide the flexibility to people to do large-scale electroporation. And then non-viral gene delivery technologies are also being worked on too. So again, being able to look at gene modification without the need for lentivirus and other types of technology is where the industry will be going. So at Thermo Fisher Scientific, our mission is really to make sure that we can provide the industry with very high-quality products that can be used in the translation of early discovery into early-phase clinical trials. We take that very, very seriously. We have a very deep quality system and a very deep quality group, as well as regulatory support to make sure that our sourcing is at the highest level so that the development of these technologies can have the confidence in the materials that are being purchased from Thermo Fisher Scientific. And our breadth of capabilities are really quite extensive too. So we can bring to bear a lot of those technologies I was talking about from a characterization standpoint to be able to make sure that your cell therapies are being tested appropriately and that you'll have the data sets that will support and allow for accelerated regulatory pathways. And of course, we have a very strong focus in this area now, very much in support of what we're seeing in the industry. So we're very happy to be able to continue developing our capabilities and really look forward to bringing them to bear and working with anybody that wants to move forward. So in summary, innovative tools to understand and drive the biology are being developed to support the cell therapy industry. It's a very active area. So we are all working very closely with the developers to understand what their needs are and then take that information and understand how we can develop more efficient tools that will allow for the development of more pure and potent and commercially viable cell therapies. So the tools that are being developed will drive commercialization through manufacturing technologies and product characterization. These technologies will ultimately reduce the cost of goods of the final products that are going to be developed. And ultimately, the development of novel and complex therapeutics will be enabled by the development in this space. So I think that's all I had for now. I think we have a fantastic follow-on to the type of things that I was talking about through Jean Loring, who's going to show specifically the application of some of these technologies and how she's using them to develop a therapeutic that is going into the clinic now. So thanks very much.